Molecular absorption spectroscopy in the ultraviolet visible region of the spectrum is a widely used technique in chemistry, biology, forensic sciences, engineering, agriculture, and many other fields. This technique allows us to quantitatively determine the concentration of many organic, inorganic, and biological species. Its fundamental is that some molecules are capable of absorbing energy in the ultraviolet or visible region of the electromagnetic spectrum. The transfer of energy to the molecule results in a decrease in the intensity of the incident electromagnetic radiation. Absorption of radiation thus attenuates the beam in accordance to the Beer-Lambert's law. Before we start explaining the fundamentals of the Beer's law, please take a minute to review some of the symbols and concepts around it, which are summarized in the following table. The Beer's law tells us quantitatively how the amount of attenuation depends on the concentration of the absorbing molecules and the path length over which absorption occurs. As light traverses a medium containing an absorbing analyte, the light decreases as the analyte becomes excited. For an analyte solution of a given concentration, the longer the length of the medium through which the light passes, the more absorbers are in the path, and the greater the attenuation. Similarly, for a given path length of light, the higher the concentration of absorbers, the stronger the attenuation. The transmittance of the solution is the fraction of incident radiation transmitted by the solution. The absorbance is related to the transmittance in a logarithmic manner as shown in the equation. Notice in the table that as absorbance of the solution increases, the transmittance decreases. To measure transmittance or absorbance, the analyte solution must be held in a transparent container or cell. However, within this cell, reflection and scattering may result in substantial beam attenuation. We can compensate for these effects by comparing the power of the beam transmitted by the analyte solution with the power of the beam transmitted by an identical cell containing only the solvent or reagent blank. A is a proportionality constant called the absorptivity. When we express the concentration in moles per liter and B in centimeters, the proportionality constant is called molar absorptivity and is given the epsilon symbol. By knowing this last constant, we can determine the concentration of a given analyte after measuring absorbance.
The spectrophotometer has four essential components. First of all, the source of light. There are two light sources, the tungsten lamp, which generates visible light, and a deuterium or hydrogen lamp, which generates UV light. The deuterium lamp gives wider and more intense light in UV region than a hydrogen lamp. Next comes the wavelength selector, for which filters or monochromators are used. A result polychromatic radiation into its individual wavelengths and isolate these wavelengths into very narrow bands. It contains an entrance slit that admits the polychromatic light from the source, a collimating device that focuses the polychromatic light onto the dispersion device, a prism or grating that resolves the wavelength, a focusing lens or mirror, and then an exit slit that allows the monochromatic beam to escape. Cuvettes hold the sample. These are optically transparent cells made of glass, silica, plastic, or quartz. Plastic and glass cannot be used for the light measurements in UV regions since they absorb UV light below 310 nanometers. Silica and quartz can be used for both UV and visible light measurements since they don't absorb UV light. And in the end, we have the detectors. These work mainly via photoelectric effect. The current is directly proportional to the light intensity. There's a measure of absorbance or transmission.
This technique can have a wide variety of applications, ranging from quantitative to qualitative analysis. In quantitative analysis, we can determine the concentration of specific analytes in a sample given the absorbance in a certain wavelength. In qualitative analysis, we can use a full wavelength spectrum to figure out the nature of a component in the sample. This spectrum is used as a molecule's fingerprint. In biotechnology, protein determination is essential for several purposes. This measurement can be done by different methods. Some of them use the UV spectrum to measure aromatic amino acids in the sample's proteins. Others use specific reagents to enable protein quantification in the visible spectrum. Absorbance is used as a means to determine the kinetics of a reaction by measuring substrate or product concentration over time. This is very useful to characterize enzymatic activity, the reaction equilibrium, the action of inhibitors or catalyzers, and the kinetic constants that best fit the reaction model. To reduce the error from other components in the sample, dual wavelength spectroscopy is used for more accurate results. Also, derivative spectroscopy mainly uses first and second order derivatives to detect small changes of absorbance over specific wavelength changes. However, higher order derivatives can also be used. This technique can be useful to reduce the error from overlapping peaks in multi-component analysis. Finally, novel in situ applications take advantage of these methodologies as a way for reactor automatization without the need to take and prepare samples from the reactor in specific time intervals.